History of DNA. History of DNA. Chromosomes are made of coiled DNA and protein. Experiments on bacteriophage viruses by Hershey and Chase proved that DNA was the cell's genetic material. DNA structure. Rosalind Franklin took diffraction X-ray photographs of DNA crystals. In the 1950s, Watson and Crick built the first model of DNA using Franklin's X-rays double helix. DNA structure. DNA two strands coiled called a double helix. Sides made of a pentose sugar deoxyribose bonded to phosphate groups. Center made of nitrogen bases bonded together by weak hydrogen bonds. DNA double helix rungs of ladder nitrogenous base, A, T, G or C. Legs of ladder. Phosphate and sugar backbone. DNA nucleotide OO equals PONCH2OC1C4C3C2 phosphate group. Nitrogenous base. A, G, C, or T. CH2. O. C1. C4. C3. C2. 5. Sugar. Deoxyribose. Pentose sugars are numbered clockwise 1 to 5 CH2O C1 C4 C3 C2. Deoxyribose Chargaff's-Rule-adenine must pair with thymine. Guanine must pair with cytosine. The bases form weak hydrogen bonds. T. A. G. C. Nitrogenous bases double ring purines adenine, A, guanine, G. Single ring pyrimidines. Thymine, T. Cytosine, C. A or G. T or C. DNA. P. O. 1, 2, 3, 4, 5. P. O. 1, 2, 3, 4, 5. G. C. T. A. Anti-parallel strands. One strand of DNA goes from 5 to 3 sugars. The other strand is opposite in direction, going 3 to 5 sugars. Question: DNA 5-3. What would be the complementary DNA strand for the following DNA sequence? DNA 5-3. Answer: DNA 5G-3. DNA 3-5. DNA and genes. DNA contains genes, sequences of nucleotide bases. These genes code for polypeptides, proteins. Proteins are used to build cells and do much of the work inside cells. Polypeptides. Amino acid chains are called polypeptides. DNA begins the process. DNA is found inside the nucleus. Proteins, however, are made in the cytosol of cells by organelles called ribosomes. Ribosomes may be free in the cytosol or attached to the surface of rough ER, starting with DNA. DNA's code must be copied transcription and taken to the cytosol. In the cytosol, this code must be read translation so amino acids can be assembled to make polypeptides proteins. This process is called protein synthesis. RNA. RNA differs from DNA. RNA has a sugar ribose. DNA has a sugar deoxyribose. Other differences: RNA contains the base uracil, U. DNA has thymine, T. RNA molecule is single-stranded. DNA is double-stranded. DNA. Types of RNA. Messenger RNA, mRNA, copies DNA's code and carries the genetic information to the ribosomes. Transfer RNA, transfers amino acids to the ribosomes where proteins are synthesized. Ribosomal RNA, RNA. Messenger RNA, long straight chain of nucleotides made in the nucleus. Copies DNA. Contains the nitrogen bases A, G, C, U, no T. Sequence of 3 bases called codon. Transfer RNA, J, cloverleaf shape. Single-stranded molecule with attachment site at one end for an amino acid. Opposite end has 3 nucleotide bases called the anticodon. Codons and anticodons. The 3 bases of an anticodon are complementary to the 3 bases of a codon. Example, codon ACU. Anticodon UGA. UGA. ACU. The genetic code a codon designates an amino acid. An amino acid may have more than one codon. There are 20 amino acids, but 64 possible codons. The genetic code. Use the code by reading from the center to the outside. Example, August codes for methionine. Transcription and translation. Transcription. The process of copying the sequence of one strand of DNA, the template strand. mRNA copies the template strand. Requires the enzyme RNA polymerase. Occurs in the nucleus. Template strand. Question. What would be the complementary RNA strand for the following DNA sequence? DNA 5G sectag 3. Answer. DNA 5G sectag 3. RNA 3 KGC AUAC 5. Translation. Translation is the process of decoding the mRNA into a polypeptide chain. Ribosomes read mRNA 3 bases or one codon at a time and construct the proteins. Occurs in the cytoplasm.